The overall goal of this procedure is to label and visualize mtDNA replication in cultured neurons. This is accomplished by first incubating neurons with the thymidine analog EDU for 2 to 24 hours. The second step of the procedure is to label the EDU that contains an alkyne with the fluorescent Oregon green 488 azide through a copper-catalyzed covalent reaction. The final step of the procedure is to amplify the Oregon green signal with a tyramide signal amplification step in order to visualize the EDU that was incorporated in newly synthesized mtDNA. Ultimately, results can be obtained that show mtDNA replication in specific subcellular compartments of neurons through the combination of click-it EDU chemistry and subsequent tyramide signal amplification.
The main advantage of this technique over existing methods like BRDU labeling is that EDU labeling is easier and milder, allowing for additional immunofluorescent staining of neuronal markers.
This method can answer key questions in the regulation of mitochondria number, such as how neurons meet changing energy loads in their subcellular compartments, including cell bodies, axons, dendrites, and synaptic terminals.
So this method can provide insight into the mechanisms underlying the pathogenesis of multiple mitochondrial-based neurologic diseases, including neuropathy and neurodegeneration. But importantly, it can also be applied to other systems where changes in mitochondrial DNA copy likely underlie the pathogenesis of the disease state, including drug toxicity, cancer, and aging.
This video will demonstrate how to label newly synthesized mitochondrial DNA, abbreviated mtDNA, in dorsal root ganglion neurons that have been grown on sterile 12mm glass cover slips in a 24-well culture plate. Dilute the 10 millimolar stock of 5-ethanol-2-deoxyuridine, abbreviated EDU, from the click-it EDU microplate assay kit 1:100 in culture medium for a 10x EDU stock. The 10x EDU stock is then added directly into the medium in each well. Incubate the treated neurons for 2 to 24 hours at 37 degrees Celsius and 5% carbon dioxide. In a fume hood, fix the neurons in 2% paraformaldehyde for 10 to 15 minutes at room temperature. Wash twice in 1x PBS for 2 to 5 minutes each wash. The fixed neurons can be stored in fresh 1x PBS at 4 degrees Celsius for up to 1 month. Prepare a humid chamber as described in the accompanying text. Use fine-tipped forceps to transfer 28 fixed cover slips, including positive and negative EDU controls, to sheets of hydrophobic parafilm ELM inside the humid chamber. Reapply 200 to 300 microliters of 1x PBS to cover the surface of each cover slip. Prepare the click-it assay kit as described in the text and the manufacturer's instructions. To label mitochondrial DNA, add 75 to 80 microliters of a permeabilization solution containing 0.1% Triton X in 1x PBS to each cover slip. Cover and incubate at room temperature for 10 minutes. Use a bulb transfer pipette with a 200 microliter tip affixed to the end to gently remove the liquid without losing cells. Use a bulb pipette to gently flood the cover slip with 200 to 300 microliters of 1x PBS to thoroughly wash out the previous solution. Wash each cover slip twice. Pipette 75 to 80 microliters of fresh 1% hydrogen peroxide in 1x PBS onto each cover slip. Incubate covered for 30 minutes at room temperature to quench endogenous peroxidase activity. Rinse twice with 1x PBS as before. Freshly prepare the click-it reaction cocktail as detailed in the accompanying text. Pipette up and down to mix, do not vortex. Post-fix the neurons with 75 to 80 microliters of click-it EDU fixative per cover slip. Incubate at room temperature for 5 minutes. During the post-fix incubation, dilute the reaction cocktail with an equal volume of click-it EDU fixative. Add the reaction cocktail to each cover slip. Cover to protect from light and incubate at room temperature for 25 minutes. Gently remove the reaction cocktail. Wash twice with diluted 1x blocking buffer. Under an epifluorescent microscope, check for Oregon green staining in nuclei. When the EDU staining is complete, begin the tyramide signal amplification, or TSA. Add 1% TSA block solution to each cover slip and incubate at room temperature for 30 minutes. Briefly spin the anti-Oregon green horseradish peroxidase conjugated antibody stock prepared 2 to 24 hours in advance of TSA. Dilute the primary antibody 1 to 300 in TSA blocking solution and invert or pipette to mix. Do not vortex. Add 75 microliters to each cover slip and incubate at 4 degrees Celsius overnight. The next day, rinse 3 times with 1x PBS at room temperature. Incubate the cover slips in the final wash for an additional 30 to 60 minutes to ensure that unbound primary antibody is removed. Prepare the tyramide reaction according to the text portion of this protocol immediately before use. Incubate the cover slips with the tyramide reaction for 15 minutes at room temperature. Wash 3 times with 1x PBS, incubating in the final wash for 30 to 60 minutes as before. Under the microscope, check for mtDNA labeling. Mount successfully stained neurons on glass slides with an anti-fade mounting medium containing DAPI. Shown here are representative differential interference contrast images of embryonic and adult dorsal root ganglion neurons that are typically used for analysis. These schematic diagrams represent the procedure for labeling EDU in mtDNA with green fluorescent signal. Mitotically active F11 neuroblastoma cells serve as a positive control and illustrate the labeling pattern of EDU that was incorporated in nuclear and mitochondrial DNA. In these representative fluorescence images overlaid over bright field, the green punctate signals show the amplified EDU signal incorporated into newly synthesized mtDNA of both embryonic and adult dorsal root ganglion neurons. The EDU labeling procedure allows for subsequent immunofluorescence staining of neuronal markers, such as neurofilament, in red. These schematic diagrams represent the procedure for labeling EDU in mtDNA with a red fluorescent signal. Mitotically active F11 neuroblastoma cells serve as a positive control and illustrate the labeling pattern of EDU that was incorporated in nuclear and mitochondrial DNA. This schematic diagram represents the procedure for labeling BRDU in newly synthesized mtDNA with either a green or red fluorescent signal.
While attempting this procedure, it's important to remember to run controls using mitotically active cells in order to check for the successful EDU labeling in cell nuclei before proceeding to the amplification steps to visualize mtDNA labeling.
Following this procedure, other methods like standard immunofluorescence can be performed in order to answer additional questions like how mitochondria DNA synthesis is regulated in specific subpopulations of neurons or neuronal compartments.
Because of its development, this technique will pave the way for researchers in the field of neurobiology to explore the regulation of mitochondrial number in culture models of both normal physiology as well as culture models that simulate impaired disease neurologic states.